Dear friends, today we will learn an introduction to algal biotechnology. So, what is algal biotechnology? Biology is what? The growth of algae using biological as well as the technical way. So, algae grow होने के लिए the basic requirement जो है वो है sunlight. Okay, this is a natural source of light. Then algae also requires CO2 for its multiplication, more multiplication process. So जो भी इंडस्ट्री से जितने भी कार्बन डाइऑक्साइड रिलीज होता है वो फिक्स करता है माइक्रो एल्गी. सो ऑल द कार्बन डाइऑक्साइड रिलीज फ्रॉम वेरियस इंडस्ट्रीज, केमिकल इंडस्ट्रीज, they are fixed by microalgae. Also, wastewater can be cleaned by microalgae. So, after all this process, the gases that are released out after the treatment will with the algae will be the cleaned gas. तो मतलब जो रिलीज कर रहा है बाहर जो गैस आफ्टर द ट्रीटमेंट वो होता है क्लीन्ड गैस. Okay, so there are some applications from this algae or the growth of algal biomass की co-firing के लिए यूज किया जाता है in green power. Okay, for the green power. Then, for the fermentation process, such as esterification, this can be used in the production of biodiesel, bioethanol, as well as biochemicals. Also, when the algae is dried, those algae can be used as a human or an animal nutrition or as a byproducts in medicines or some high valued molecules. So this was an overview about the algal biotechnology. Okay, so Q algae. Why only algae is generally used for all this purpose? Why? The main two reasons behind this is the strain develop improvement and production efficiency. Okay, the first reason is that strain improvement. The strain improvement is applied to develop an increase in heat tolerance, salt tolerance, or light harvesting ability, means the sunlight and the digestion activity. Also, it has a very good reason for the production efficiency, like a new approach has been developed for efficient CO2 delivery as well as a new model for scale up low energy ultrasonic algae harvesters. Okay. Now, how to use it? So let's see how we can use this algae. For this, we need to know how the algae is based to bioresource cycle. So this is a natural bioresource, means this is a natural basic raw material from where the algae can be collected. तो यहाँ से algae collect करके उसको पहले identify करेंगे using the microscope. Then this algae, the particular species of algae can be extracted. Okay. और उसको कल्चर करेंगे हम डिफरेंट पॉन्ड. सो दैट पॉन्ड इज नोन एज व्हाट? एल्गी कल्चर पॉन्ड्स. Okay, now for the growth of algae, the sunlight is an important part. So sunlight is required. Also CO2 is required. So CO2 comes from different industries. The gases released from industries can be used, okay, by the algae. And on the one way it cleans that the air also, okay, from controlling the pollution. और algae culture ponds में क्या required रहता है? Mineral nutrients. So the mineral nutrients are in the ratio of NPK, nitrogen, phosphorus and potassium. Okay, so you could see here the anaerobic digester. So you could also get mineral nutrients directly from the anaerobic digester, where the anaerobic digester is used for the production of the biogas such as methane. सो यहाँ से मीथेन भी मिलता है और CO2 भी मिलता है. तो वो जो CO2 है और डिफरेंट मिनरल न्यूट्रिएंट्स है वो डायरेक्टली कहाँ यूज कर सकते? एल्गी कल्चर पॉन्ड्स. नाउ this methane produced from the anaerobic digester are used by the society. Means people use biogas for cooking and various other purpose. और जो algae culture ponds में जो algae biomass जो बनता है that are used as a bioresource feedstock, where this bioresource feedstock is further processed for bioregional processing. Means bioregional processing के लिए use करके क्या form होता है, feeds, fuels, fibers, ये सारे चीजें form होता है, and this all things they are utilized by the society. So, and this bioregional processing, that process can be extended back to the anaerobic digester. So, this completes the cycle known as bioresource cycle based to algae. So, in this, we could use algae for the nature, for the environment, and for the society. Now, we will see which are the culture techniques of microalgae, how the algae could be cultured. So, algae culture techniques can be based or divided into 4 different ways, like based on place, based on container, based on sterility, based on production, etc. So, based on place, it can be divided into two types, techniques like indoor and outdoor. Based on container, it can be divided into open and closed culture. And based on sterility, it can be divided as axenic and scenic. And based on the production, they are divided into batch, continuous and semi-continuous culture techniques. So let's see brief overview of all these cultures. Like the first one is indoor and outdoor culture. So you could see the picture of the indoor and the outdoor culture. The main thing in indoor culture is that indoor culture allows to control illumination, temperature and competing with particular species of algae. But in outdoor culture, it is difficult to grow specific algal culture for extended period of time, as the sunlight is not available, the light source is not available for 24 hours, so it may extend for particular periods. Next is that open or closed algae. So you could see that the picture showing the open or the closed algae. बेसिकली ओपन कल्चर मतलब क्या होता है, इट इज एन अनकवर्ड पॉन्ड्स और टैंक्स. ये इंडोर भी हो सकता है या आउटडोर भी हो सकता है. सो देयर आर द चांसेस दैट दिस ओपन कल्चर्स मे बी मोर कंटेमिनेटेड, बट इन क्लोज्ड कल्चर वेसल जो कि किस में होता है, ट्यूब्स, फ्लास्क, कार्बोइड्स, बैग्स, ये सारे प्रोसेस में होता है, ये सारे कंटेनर्स में होता है, और there is a less chance of contamination in the closed culture techniques. The next one is axenic or xenic culture technique. तो यहाँ पे पिक्चर में देख सकते हो कि axenic means you could see it's totally packed, means it is totally sterilized. The meaning of axenic is that under the sterile condition, culture, the algae culture is done under the sterile condition. So axenic means sterile and means it is free of foreign or any other organisms such as bacteria, fungi, etc. Means the strict sterilization is done for the glasswares, for the medium, for the vessels that are used for the culturing techniques. और ये क्यों करते हैं स्टेराइल कंडीशन या स्टेराइल टेक्निक क्यों यूज करते हैं, टू अवॉइड द कंटामिनेशन. बट if you go for the practical purpose, for the large scale or for the industrial and commercial scale, it's not practically possible because sterilization at such large scale, it's very complicated. Until now we have done all the different algal culture techniques like closed culture system. जैसे कि closed culture system मतलब contamination कम होगा, means it is also known as axenic culture. If it is open culture system means there's a chances of more contamination, मतलब ये होगा non axenic culture. तो ये सारे different types of culture techniques हैं. They can be operated in 3 different modes like batch mode, semi continuous mode and continuous mode. ओके, तो हम इसमें अब देखेंगे सारे टेक्निक्स या सारे ऑपरेटिंग मोड्स को, कोई एडवांटेजेस एज वेल एज डिसएडवांटेजेस होगा। तो हम देखते हैं कि क्लोज कल्चर सिस्टम में क्या होता है कि क्लोज कल्चर सिस्टम इज एक्सपेंसिव, so this is one of the disadvantage, and but it can be controlled, means all the temperature, pressure, all the conditions, light conditions can be controlled, and there is a less chance of contamination. But in the case of open culture system, it's cheaper, means as it is open, it's done in open space, it is cheaper, it is less controllable, means it cannot be controlled easily, and so it has more prone to the contamination, मतलब contamination होने के chances ज़्यादा है। अब हम देखते हैं कुछ एडवांटेजेस और डिसएडवांटेजेस ऑफ बैच मोड, कंटिन्यूअस मोड एंड सेमी कंटिन्यूअस मोड। तो बैच मोड में एडवांटेज है, इट्स मोर रिलायबल एंड इजिएस्ट टू मेंटेन, बट द डिसएडवांटेज आर इनकंसिस्टेंट एंड दे आर लेस एफिशिएंट। सेम वे इन सेमी कंटिन्यूअस मोड वो भी इजियर टू मेंटेन है, but the disadvantage is that it is less reliable and it is somewhat consistent. तो continuous mode में जो advantage है, it has high efficiency as well as high production, but disadvantage is same, like it's inconsistent and is less reliable, and it is also difficult to maintain. ओके, सो अब हम देखते हैं different biotechnological applications of microalgae. So where this microalgae, the produced microbial biomass, can be applied, can be used. So it can be used for food and dietary supplements, right? For now, as a biofuel, it can also be used as a biofuel, such as biogas, bioethanol, biodiesel, and biobutanol. Also can be used as a feedstock, okay, for the cattles, fish, okay, different animals, etc. It can be used as a fertilizers. So these are the best organic fertilizers that can be used for the crops, for the agriculture purpose. Also can be used in the production of bioplastics. So those are biodegradable plastics, okay. Also, the algae, the microalgae can be used in the production, the formation of different cosmetics, okay, that can be used, that can be used for the skin, hair, etc. Now, let's see how this algae came, okay, into which generation. So there was, हम कंसिडर अगर करेंगे फर्स्ट जनरेशन, सेकेंड जनरेशन, थर्ड जनरेशन, तो कौन से जनरेशन में एल्गी का इन्वॉल्वमेंट हुआ, स्टार्ट हुआ। तो फर्स्ट जनरेशन में फीड स्टॉक जो यूज करते थे वो था कॉर्न, व्हीट, ऑयल सीड्स, ये सब, और प्रोडक्ट्स जो बनता था इसमें से वो था बायो इथेनॉल, बायोडीजल, वेजिटेबल ऑयल, बायो ईथर, सॉलिड बायोफ्यूल्स, एक्सेट्रा। नाउ लेट्स सी इन द सेकेंड जनरेशन जो फीडस्टॉक्स यूज करते थे वो था नॉन फूड क्रॉप्स, इनएडिबल वेस्ट प्रोडक्ट्स, सम सिट्रस पील्स, सॉडस्ट, एक्सेट्रा। और जो प्रोडक्ट्स इसमें से जो बनता था वो था सेल्यूलोसिक इथेनॉल, बायोहाइड्रोजन, बायोमीथेन, फिस्कर, ट्रॉप्सिकल, डीजल, माइकोडीजल, एक्सेट्रा। तो ये था पूरा ओवरऑल फर्स्ट एंड सेकेंड जनरेशन। अब हम अगर कंसिडर करेंगे थर्ड जनरेशन का, तो थर्ड जनरेशन में यूज करने लगे एल्गी, दैट are different types of microalgae and macroalgae. So microalgae में जो ज़्यादा यूज करते थे वो थे स्पाइरुलिना, बोट्रियोकॉकस, ब्रॉनी, क्लोरेला वल्गैरिस, एक्सेट्रा। तो ये जो मैक्रो एंड माइक्रो एल्गी यूज करके डिफरेंट टाइप ऑफ एल्गिल बायोफ्यूल्स, मतलब जो मेन कॉन्सेंट्रेशन था प्रोडक्ट का वो था फ्यूल, एल्गी फ्यूल को प्रोड्यूस करना। तो ये था मेन बेसिकली द एल्गी केम, okay, into the involvement in the third generation. Okay, so this was a brief introduction, okay, towards the algal biotechnology. So we'll further do the detail, okay, of different algae types, the microalgae species in detail in the next videos. So thank you for watching. Please do subscribe, like and share if you like our video.